It's really my pleasure to introduce Dr. Alex Bick. So Alex did his MD and PhD at Harvard in John and Cricket Seidman's lab. Then he did his internal medicine residency at MGH and a postdoc at the Broad with Sake Kathareson before moving to Vanderbilt, where I had the pleasure of meeting him. And he's now risen up at Vanderbilt to be the division director of genetic medicine and clinical pharmacology. And he holds the Edward Stallman Chair in Medicine, as well as sees patients in the genomic and therapeutic clinic. And his work is focused on both germline and somatic mutations, how they impact and interplay on cardiovascular health, as well as really across human disease. And that's been recognized with multiple honors, including from ASCI, the Selden Smith Award recently, for pioneering research. And Alex also just a really great mentor already in his young career, sent a lot of PhD students and postdocs off onto their own successful trajectories, and great friend. So really my pleasure to introduce Alex to give us grand rounds.
Thanks everybody for showing up in person. It's like always fun seeing that people have legs. So this morning I want to start by taking you back a bit, about 10 or so years. Imagine you're a DeLorean. So, what was happening here in LA in 2014? Well, the LA Kings, won the Stanley Cup in double overtime over the Rangers. And I grew up in New Jersey, so I actually remember this game. And I think the LA Times had the headline that UCLA football really had come into its own after pummeling USC. And now Vanderbilt's actually a very big football school now, which is something I'm learning a lot about. People are wandering around with these Google Glass things, which have really not caught on. But in hematology things were looking a little bit different, and this was because we, for the first time, had actually found this thing called clonal hematopoiesis, which was a pre-blood cancer mutation. That for time immemorial, we knew that colon cancer develops from colon polyps, and breast cancer develops from precancerous lesions, and so everyone had always said, well, blood cancer must have a pre-blood cancer as well, but no one had ever been able to find it before. In 2014. Because we started doing large-scale sequencing, two different labs, Van Ebert's lab and others, we're able to identify this precancerous mutation. And again, this makes a lot of intuitive sense if you think about the biology. So we're all born with hematopoietic stem cells. All of the cells in our body, as they grow and divide, acquire mutations. Most of these mutations don't do anything. But once in a while, you can have a mutation that occurs in the wrong place at the wrong time. And now all of a sudden that gives an advantage to the blood stem cell, and now it can start growing. And it can grow faster than its other stem cells and start taking over. And so today I'm going to tell you a few different things. One, talk about what is clonal hematopoiesis. Talk a little bit about the cardiovascular epidemiology, since this is cardiology grand rounds. Talk about some of our work to understand why CHIP clones expand, and then how, once they expand, do they cause cardiovascular disease, and end up with some thoughts about what we're doing at Vanderbilt and others are doing elsewhere for patients who are found to have clonal hematopoiesis. Let me see if Michael can tell us how to find this top bar.
You go to more,
Yeah.
and hide the floating controls.
Excellent. Thank you. Wow.
So Vanderbilt's moved over to Teams, which makes me completely Zoom illiterate. So there are many different kinds of mutations that can cause this kind of clonal expansion. You can have single nucleotide changes in genes that cause myeloid blood cancer. We call that CHIP. You can have mutations that involve gains or losses of entire chromosomes or segments of chromosomes. And all of these different kinds of mutations give rise to a clone, but they actually have different health and disease consequences. So for purposes of today, I'm going to focus on talking about CHIP, again, one side of the umbrella. But these other things are also very interesting too. We'll save that for conversations later in the day. And so CHIP has a technical definition now, where it involves having a mutation in a myeloid cancer gene, having that clone grow in size such that more than 2% of the peripheral blood DNA has that mutation. And that they have this mutation and they don't have some other blood abnormality that might explain why they would have a mutation. So it's truly a pre-malignant lesion. There's a related term called CCUS, or clonal cytopenia of undetermined significance, which basically means you have a mutation and you have a cytopenia, but you don't yet have some changes in your bone marrow that you might call it myelodysplastic syndrome or myeloid leukemia. So these things are sort of exist on a spectrum, and hematologists, just like cardiologists, love acronyms. So this is the alphabet soup we're left with. We have made remarkable discoveries in CHIP because we sequence patients' blood. So if most of our sequencing came from people's cheek swabs, we would not know anything about CHIP. We could tell you all about the oral epithelium. And when you do sequencing, this is a picture of a next generation sequencing screenshot. Basically, each of the gray bars is one read. And we're able to identify that this particular deletion, this one base deletion, occurs in 3 of the reads. And because 3 is not half, we can infer that it's not inherited. So if mom or dad passed on this mutation to this patient, half of the reads should have this deletion. And this is again the intuition that has sort of powered this field. Today in the clinic at Vanderbilt, we get referred to our cardiologists a fair number of these patients, probably about 5 to 10 a week, because our cancer clinic is doing a lot of sequencing. And as part of their sequencing for breast cancer or for lung cancer or for pancreatic cancer or whatever, they're sequencing these genes and they're finding mutations in the blood that are not in the tumor that they're looking for. And we're getting on clinical reports this report back that the patients have clonal hematopoiesis. And so we've built a partnership between the cancer folks and the cardiology folks so that when the cancer clinicians see this, they know that this might be something that's important and send them over. But we know a lot more about this because of global biobanks, including what you guys are building here with Atlas, that most of these biobanks around the world have been from blood-based DNA. And a very small number of them have been from saliva. But even saliva, the DNA in saliva is actually mostly blood cells. And so we're able to use these hundreds of thousands, millions of people around the world who have DNA to find out, do they have CHIP or not? And then we can use linkages to their health records to figure out whether this matters. We've developed fancy pipelines to do this, which is all to say that it's not as easy as it looks. And it's also not perfect. So when we look at a data set where we have an exome sequence or a genome sequence, and then we go back and we sequence it much more deeply, we're able to see that we're actually missing a whole lot of CHIP. But for people who have big CHIP clones, about half of them actually, in fact, do have CHIP. And so this is, I think, something that you guys are looking into in Atlas, is that even if you have CHIP in Atlas, probably half of those people, it's just a sequencing artifact. Which is all to say that I'm going to show you a lot of epidemiology, and half of it is wrong. We just don't.
Is that sequencing based on blood? So, okay, maybe you're going to get to it, but can you do this from liquid biopsies too?
Yeah, so in liquid biopsies, a lot of the DNA in it is actually from blood cells. You can actually do it from plasma as well. And this has been a real pain in the neck for Grail and these other big liquid biopsy companies. Because they find a P53 mutation and they're not sure, is it pancreatic cancer, is it clonal hematopoiesis? So they put a lot of effort into trying to figure this out. And, I'm not sure if they've solved it yet. Because we have these huge biobanks, we're able to look, with age, who has clonal hematopoiesis. And very commonly we report things like, oh, about 10% of people in their 70s have clonal hematopoiesis. And that's actually a gross underestimate. It's probably closer to 20 or 30% of people. And that's because of what I just showed you, that we're using exomes or genomes, which are not actually very good at detecting clonal hematopoiesis. But the flip side is that it's free. So free is good when you're a research faculty. When you're a junior fellow, you might not care as much about money, but when you have to actually pay for the bills, you care a lot about it. And so because we have essentially free access to about 1 million people's genomes, we've been able to characterize CHIP. And one of the surprising things is that there are many different genes that could cause these clonal expansions, but there's really only a very small number that actually do in practice. And you can see that it's not random. About half of people have a mutation in a gene called DNMT3A, about 20% in TET2, 10% in ASXL1, and then all of the genes that classically you think of as cancer genes, P53, KRAS, IDH, those are actually really, really small contributors to CHIP. Small but important because for some of these we have targeted therapies, but just in terms of the global landscape. And then the other thing about CHIP that again is always puzzling is that you think about it, you're like, okay, DNMT3A and TET2, what do? Well, DNMT3A takes methyl group and puts it onto cytosine. And then TET2 is also involved in methylation, but exactly in the opposite direction. It takes that methyl group and basically turns into hydroxymethyl, which is the first step of removing it from cytosine. So everyone who loves to say like, oh, CHIP, it's all about methylation, we actually have no idea what or how. Science is always more complicated. So in the 2014 papers, again, they were looking for a pre-blood cancer. They found a pre-blood cancer, and sure enough, CHIP was associated with blood cancer. Many people have replicated this finding across many different data sets. When we look in UK Biobank, we're able to bring a lot more samples to bear on this question. And one of the observations is that the bigger the CHIP clone you have, the worse it is for the patient. So if you have a clone that's a small clone, not great. You're twice the risk of getting blood cancer, but it's not 27-fold increased risk. The sort of more surprising thing that came out of these 2014 papers was that patients with CHIP were at increased risk of dying. And it was about a hazard ratio of about 1.4. And again, this too has been replicated by us and many other people in other data sets. And the thing that was really puzzling about this observation is sort of why were patients with CHIP dying? Because myelodysplastic syndrome, myeloid leukemia, these are deadly, but they're really, really rare. And so there aren't really enough people who are getting myeloid cancer to explain this hazard ratio. So there's a lot of puzzling, like how does CHIP increase mortality if it's not through blood cancer? And there was this throwaway sentence in the discussion of Ben Ebert and Sid Jaiswal's 2014 New England Journal paper. It said, well, maybe it's related to increased risk of coronary heart disease or ischemic stroke. They had done some exploratory analyses and found a couple of people who this had happened in. And this was what has set off this now sort of 10-year journey in CHIP and cardiovascular disease. This is me when I defended in the Seidman lab. They gave me this T-shirt, which I still have. My daughter was asking me about it the other day, like, Dad, why do you still have this one? And I got involved with CHIP research because I was the person who was working on sequencing data from Framingham Heart Study. And so Sid Jaiswal and Pradeep Natarajan wanted to explore this signal further. And they needed someone who had access to these data sets. So, that was my contribution to this. But basically, we found in 2017 that indeed in other cohorts that patients with CHIP did have increased risks of coronary artery disease. And that, this was also the first time we were seeing perhaps there's some differences between if you have JAK2 CHIP versus some of these other genes, your risk might be different. The model at the time, both work from Sid Jaiswal, work from Jose Fuster and Ken Walsh, was this sort of mouse model suggesting that CHIP could increase atherosclerosis by leading to increased inflammasome activation. They were able to replicate this in a mouse model, and many labs around the world have found this mouse model to replicate, which is now reproducible science is now a very big buzzword. And much of mouse science is not, and this model happens to work pretty well. The broader cardiovascular epidemiology community then jumped in and tried to figure out, is it primary prevention? Is it secondary prevention? Where is the risk of CHIP and heart disease? I worked with the TIMI study group to look at a number of their clinical trials. And one of the interesting observations is that at least in the clinical trial population, we do a really great job of adjudicating, what kind of events people actually had. Seems like a lot of the risk is actually patients who have not really had an event, and that CHIP in sort of the secondary prevention population seems to be a lot less of a risk factor. And why that is this a power issue? Is it related to the fact that these patients are on a lot of standard of care medicine? We're still puzzling over. The other interesting post hoc analysis that I did not contribute to, although I did review, so I had a small contribution, was this reanalysis of the CANTOS clinical trial data, where investigators at Novartis basically stratified people by whether they had CHIP or whether they didn't have CHIP. Overall, the CANTOS trial was positive, that being on canakinumab was associated with like a 15% decreased risk of MACE. And if you look in patients without CHIP, there's basically no benefit. The hazard ratio is like 0.93. But then when they looked in patients with TET2 TRIP, all of a sudden the hazard ratio was enormous, that there was almost like a twofold increase in benefit from this very small population of patients being on this very targeted inflammatory therapy. And so this requires replication prospective trials, and there's a lot of interest and a lot of discussion in doing these kinds of trials right now. And I'll talk about that towards the end. There was also a detour. There's a lot of interesting questions about whether CHIP causes heart disease or whether heart disease causes CHIP. I'll say that it's a good question. And Jose Fuster, working with Valentin Fuster, actually had data to answer it. And the way they were able to answer it was by having multiple time points of both subclinical atherosclerosis and imaging, and multiple time points of CHIP. And basically what they found was that if you had subclinical atherosclerosis, that did not increase your risk of developing a CHIP mutation. Whereas if you had a CHIP mutation, it did increase your risk of developing subclinical athero. And I say that just because there's still questions that sometimes we get about which came first, the chicken or the chip. And this has led, the really great thing about science is once people develop assays, develop approaches, and show you how you can do these in biobanks that are free, there's been really an explosion of this kind of research. So really across cardiovascular disease, there's now sort of new understanding of different genes that cause different forms of heart disease. And I think what's another just important thing to note is that different genes do cause different degrees of different kinds of heart disease. So it's not just the fact that you have a clone, that whether you have a TET2 clone or whether you have a DNMT3A clone or a JAK2 clone, they lead to different outcomes. And in Raditz et al. In 2025, in a series edited by Dr. Stein, over here, Michael did a great job of sort of saying, what is the lay of the land in terms of different forms of heart disease, different genes, and what kinds of risk are we talking about? Also, I guess this was during intern year that you did this. I don't remember when.
Review process.
Yeah, the review process for this paper took 5 years. Yeah. Which wasn't the review process. Part of it was the collaborators doing the mouse experiments that the reviewers wanted, which took 2 years, which was not Michael. But Michael was very interested in the context of aortic valve stenosis. Found some interesting human epidemiology suggesting that patients with CHIP, particularly with TET2 CHIP, more so than DNMT3A CHIP, are at increased risk of aortic valve stenosis.
Is there a sex difference?
Is there a sex difference in
just various kinds of CHIP?
There's there is a very small sex difference. So women are very slightly protected from getting CHIP. Why that is, we're not sure. And whether that, there's some thought that maybe it's related to hormones. There are other thoughts that maybe it's actually related to a different kind of a clone. So men have Y chromosomes, but men also lose their Y chromosomes.
Yeah.
And so it may be something about the clonal competition of loss of Y versus CHIP might be doing it. So anyway, this is the mouse experiment that took 2+ years, where basically you can take a mouse, and it's very easy to give a mouse CHIP because you take a mouse that has a TET2 mutation and you do a bone marrow transplant. And again, people in the atherosclerosis unit here, I think, pioneered this kind of an experiment probably in the 80s. And when we do that in aortic valve stenosis, the reviewers are very happy to see that you can in fact induce increased calcification at the valve leaflet. This should be coming out sometime soon. But then the question is why, and I thought this was pretty cool. So the question is like, these CHIP mutations are in blood cells. So how is it that the blood cells are interacting with the calcified valve leaflets? They're not. Are they integrating? And what we showed in vitro is that you could just take the supernatant of macrophages that have TET2 mutations, and that in and of itself was actually was sufficient to induce calcification. So I'll show you a different story later where it's actually the macrophages themselves are probably more important. Here it's actually just the evil humors. And this has not been just limited to cardiovascular disease. There's a very active area of pursuit in kidney disease. There's some interest in liver disease, and increasingly auto-inflammatory diseases of sort of any kind, vasculitis, etc., are all sort of linked to CHIP. So this is sort of the landscape of CHIP circa today, and I suspect that there will be more and more body systems added as people keep doing more sequencing. Across pretty much every single one of these diseases, bigger clones are worse than smaller clones. And so one of the early ideas that my lab was pursuing is like, well, mutations happen. We can't stop mutations. If we could, that would be really cool. Would prevent a whole lot of cancer. But maybe we could take a small clone and keep it a small clone rather than letting it progress to a big clone. And so since bigger clones have worse outcomes, we thought that perhaps faster growing clones are probably worse than slower growing clones, and we started trying to identify how do we change growth rate. And so measuring growth rate is actually relatively easy because it's just a blood sample, and from the blood sample you can directly measure how big the clone is. The problem is that having two of these blood samples multiple years apart. And the reason for that is that everybody, including Atlas, your biobank, you have one blood sample, and then you say, it was really costly to get that one blood sample. The DNA probably doesn't change, so why do I need a second one? And then CHIP came along, and now people realize that the blood actually does change. So we at Vanderbilt, we developed a relatively cheap assay, which Priscilla and Michael, others have used for their studies. And I think there's some exciting efforts to try and stand up something like this here at UCLA. But until your assay exists, we're open for business and we're happy to help. And so how do we overcome this problem of biobanks only having one time point? Well, at Vanderbilt, our biobank has a small fraction of the biobank that has two multiple samples. And the reason for that is actually, I thought it was an accident when I first came to Vanderbilt, but actually it was intentional. Because back when they started setting this up in 2005, you needed like micrograms of DNA to do genetic assays. And you would deplete your genetic DNA relatively quickly. And so once they had less than a microgram, they got a second sample just in case. And now that our assays need nanograms of DNA, not micrograms of DNA, we can take advantage of the fact that they never threw out the first sample. So Tara Mack was my first PhD student, and working with Yash Prasad, who's a very talented MSTP. basically went and looked within BioView, screened about 3,000 patients, found about 700 who had mutations at 2 time points, and then we're able to calculate the expansion rate. And the data looks shockingly messy. So in this plot, each line is a person. And what you see, and these are all people who have the same gene, mutations in ASXL1. Some of these clones are growing really quickly. You see that by the lines, the red lines that are going up. Some of these clones are barely growing at all, these gray lines. And some clones are actually shrinking, which was shocking because, again, in this model, everybody, you have a pre-cancer, it just persists and evolves and then turns into cancer. And that was true actually not just of ASXL1, but actually of almost all of the CHIP genes. Some people's clones were growing faster than others. Some clones, even with the fastest growing genes, like the splicing factors, were not, there was 1 or 2 people whose clones were not growing at all. And so this was really exciting because now we actually had some variation. And as a human geneticist, if you have variation, you can start to study it and try and understand what's causing the variation. And so the driver gene is a major determinant of the growth, but it's, it doesn't tell the whole story. In BioView, our biobank, just like Atlas, your biobank, the data is linked to electronic health records. And so one of the first things we were looking at is, can we identify medicines that patients were prescribed for other reasons that are associated with changing growth rate? And, it was a small study. It's all very exploratory. There's many, many confounding factors. Why was a patient, put on colchicine? And, we did our best to match it the best that we could. But colchicine was actually the top hit of patients on colchicine had clones that grew more slowly. And so we kind of thought that was exciting. But again, p-value of 0.02 doesn't get geneticists that excited. What really gets people excited is replication. And this just came out a couple of months ago, where looking in both of our the big using colchicine for prevention studies, both in Ledoco and in Holcott, they both have basically the same result. One is published, one's been presented in abstract form, where patients who are on colchicine, particularly patients with TET2, grow more slowly. And so, again, now having EHR data, retrospective reanalysis of RCT data makes us really excited to look to see whether this is true.
If there weren't a zillion stem cells, could it just be stochastic? If there are a limited number of pluripotent stem cells, right? Then maybe there isn't any. Maybe it's just a chance.
So in terms of the sort of random growth and shrinking, I think that's one theory, is that there's, it's a small population. There's fluctuations that happen in blood counts randomly, and maybe that's contributing. I think that, when you have observations like this, where there's a medicine that you give people that can change the trajectory, though, that's when it gets, sort of medically interesting.
Oh no! I think that's interesting. Yeah, I thinking about all the noise.
And so we didn't leave it here. We kept looking for other drivers.
Sorry, real quick, just to kind of follow up on what Jake was saying. So if you go back 2 slides and you basically the stochastic aspect, could that also be the size? Because at the beginning you said that it was basically like the size of the mutation was associated, I guess, with how bad that was going to be. Did you normalize this on size at all, or are all these kind of all different size mutations?
Yeah, so we've this plot is normalized on size because it's looking at change in a person's fraction, but we've also done stratified analyses and I think one of the observations that, to your point, is that once a clone gets to be a certain size, you're not getting rid of it. And so once a clone gets to be 10% clone, it's going to stick around. Some of these clones that are small clones, 2%, 3% clones, actually can disappear. And we're seeing that in some of Priscilla's HIV studies. So yes, there is some amount of what happened to this clone? Did it just peter out because of stem cells, just like every other cell type at some point will have the nest.
Just like Cantos, did the presence of CHIP impact the response to colchicine?
So neither group has reported that, and so I don't know whether that's a future paper or whether they didn't observe it, so they didn't report it.
And then, has anyone looked at AS progression in the setting of colchicine in any of these?
Not that I'm aware of. So we've been really interested in trying to figure out how can we use large human cohorts to figure out what affects growth rate. And so we know how old a person is when their blood is gone. We know how big the clone is. We can directly measure that from the blood. But the thing we don't know is when the clone was born, or how old is the clone, not how old is the person. And so we developed a cool approach to try and solve this problem. So if you were in Boy Scouts or Girl Scouts, my daughter's a Girl Scout, you know that like if you're hiking through the woods, sometimes you come across trees that have fallen down or been cut down in your backyard in this case. And my daughter, she did this spontaneously, believe it or not. She went and was counting how many rings was on this tree and was able to tell me that this tree was 89 years old. She counted 89 rings. Hematopoietic stem cells actually do the same thing. In one of my very first slides when I was telling you that we acquire mutations randomly over time, that means that the older a stem cell is, the more mutations it's accumulated. And so you can use this idea to figure out how quickly a clone is growing from just a single time point. We called this approach PACER, something I developed with Sid Jaiswal at Stanford and Josh Weinstock, who's now has his own lab at Emory. But imagine that you have two people, they're both the same age. They have clones that are making up the same fraction of their blood. But one person has 3 passenger mutations. There's 3 rings on the tree, and 1 driver mutation, and the other person has 7 rings on the tree. You know that that clone in person 1 was born earlier than the clone in person 2. And so if the clone was born earlier and grew to be the same size, and as the person's the same age, and the other clone was born later, you know that in order to become the same size, it must have gotten there by growing faster. And so with this, again, approximation, we can go from just measuring whether or not you have CHIP or don't have CHIP with a single time point to how fast the clone was growing. And so we can then take that into thousands, hundreds of thousands, now up to 1 million people, and estimate for each CHIP clone, what their growth rate is, and then do a genome-wide association study. And so our first stab at this, there was a single peak, which is great when you're writing up GWAS papers. Now we have the problem that there's 300 peaks, and then you're not sure what to write about other than it's genetic. And so it was a variant in an enhancer near TCL1A, in the promoter of TCL1A. And so this was just an estimate. So we went and found some smaller cohorts where we did actually have measured growth rates, one from the Women's Health Initiative, one from BioView. And, again, across multiple cohorts, we found that having more copies of the alternate allele at this particular locus led to slower growth rate. So we're like, this is cool. How is this happening? What is TCL1A doing? And it was really discouraging because we actually looked it up in the genome browsers, and there is no chromatin accessibility, no gene expression at all of TCL1A until we looked at the chromatin accessibility and the gene expression in patients who had a TET2 mutation. And that TET2 changes DNA methylation, changes chromatin accessibility. And patients who have a TET2 mutation, all of a sudden, this promoter region was accessible. And so, again, using many of the tools that we all know and love, we actually would have and did completely miss this until we started understanding the context a little bit more. What's interesting is we then found hematopoietic stem cells from patients who either had 0, 1, or 2 copies of this allele. and then added the TET2 mutation to the hematopoietic stem cells. And you can actually show that if they have the reference allele, this GG allele, TET2 does open up the chromatin accessibility. If you have one copy of the allele, it opens it up a little bit. If you have two copies of the allele, for whatever reason, TET2 really has a much harder time opening up this promoter region. So, TET2 is sort of like a cheat code. It sort of helps turn on this gene that's not normally turned on in hematopoiesis, but having this genetic variant sort of prevents that from happening. We did experiments in vitro and in vivo where we're able to show that T cell 1A is actually necessary for TET2 clonal expansion, that if you both create a TET2 edit and you knock down T cell 1A, the clones really don't expand very much. And it's also sufficient that if you just overexpress TCL1A, which again is not normally expressed in hematopoietic stem cells, it can lead to significant growth. And this is true. And so, across all of these experiments, we showed that PACER worked, but more importantly, we found a target. Is it a good target or a bad target? I don't know. That might change, the growth of clonal hematopoiesis in patients with TET2. So Michael was wandering aimlessly in fourth year med school before I found him. We wanted to build on this, looking at a few different things. One of which was looking at DNA methylation itself is a risk factor for growth rate, was one of the observations. But you can also use this tool, and Michael did in this paper, to find a number of other targets, basically proteins that are circulating in the blood that seem to change clonal growth rate. So that's what we've done to try and get at sort of the root issue of CHIP. We've also been very interested in understanding, why and how is it that CHIP causes atherosclerosis and vascular inflammation. And I'd say, and a lot of this work has been led by Brett Heimlich, who is a cardiology fellow in my lab and is now transitioned to assistant professor. And when the dust settles, we'll have a great startup package, but I don't clear exactly what that would So he had this idea. He said, well, Alex, this is really simple. And Priscilla wrote a great grant that said the same thing. Why don't we just do single cell sequencing and find out the difference between the mutated cell and the not mutated cell? And again, this sounds like a great idea. It took him 3 years to try and figure out a set of technologies to put together to make this sort of work. And I say sort of work because even by putting all these technologies together, it only works about a third of the time. But what he found was that you can use mutations that are occurring in the mitochondria. So sorry, let me get back to the problem. Problem is that DNMT3A, TET2, these other CHIP genes are not expressed in mature blood cells because they set the DNA methylation and then they turn off as the cell differentiates. And even when they are expressed, they're expressed at very, very low levels. And so single cell RNA sequencing will never pick them up because there is no RNA. And so he needed to find a way to identify these cells. And one thing that does produce a lot of RNA that we don't want is the mitochondria. So when you do single cell RNA sequencing, you try and throw out a lot of mitochondrial DNA reads, RNA reads. And what he was able to do using a different single cell technology was identify which mitochondrial mutations co-occurred with the CHIP mutation. And then once he knew that, then he could do normal single cell sequencing and mitochondrial sequencing at the same time, and then put all those things together to say, therefore, if it has the mitochondrial mutation, it must also have the CHIP mutation. What he found with this approach was actually pretty illuminating, though. And again, in retrospect, there are probably easier ways to do it, but that's the benefit of hindsight in science. That if you have a patient who has, let's say, a 10% TET2 mutant CHIP clone, it's not that every single blood lineage has 10% mutations. It's that CHIP mutation leads to a myeloid bias. And so you get certain subsets of cells, like CD14 monocytes, where even though 10% of the blood overall is mutated, 90% of the monocytes are mutated, the CD14 monocytes. And a very small fraction of the T cells are mutated because it leads to lineage bias away from the T cells. And so this is why even if you have a 2% or 3% or 5% or 10% clone, you can actually get systemic effects. He was able to use this to look at different inflammatory pathways. One that he's followed up with in a mouse model is a gene called MIF, and that's all fascinating. But he wanted to go deeper and say, well, what's actually happening in the vasculature of patients, not just the blood. That's where cirrhosis happens, and so they said, well, how are we going to get heart tissue? It's hard to get heart tissue, and it's even harder if only a fraction of them have mutations in CHIP. And they said, well, what do we have a lot of in Tennessee that you guys don't have any of in LA? And that is fat. We have a lot of adipose tissues that patients are very happy to part with. And so adipose biopsies actually provide this amazing window into cardiovascular biology, because mixed in with the fat are vascular smooth muscle cells and endothelial cells. And again, colleagues actually in the HIV world, John Coates, Sally Wanjala, they figured this out a long time ago. And we said, well, now we can actually see what's happening, both in the vasculature and in the blood, without eating a piece of people's aorta. So we did single cell sequencing across a set of patients with CHIP, and both in their blood and in their adipose tissue. And the cool thing is that there are now computational tools that you can use to understand what are some of the different signaling interactions happening between the blood cells and and from this set of interactions between integrins on the macrophages, integrins on the endothelial cells, we're able to identify a subset of specific pathways that we thought seemed to be playing a role. Interestingly, they were different between TET2 and DNMT3A. There are some things in common, there's some things that were different. And then that can lead to a specific hypothesis from these patient samples that, if you take myeloid cells that have a CHIP mutation, do they roll differently over endothelial cells? And sure enough, the answer was yes, that patients with TET2 or DNMT3A monocytes seem to be more sticky. They seem to move a little bit less over these layers of endothelial cells in culture. And then we've been doing experiments that you can actually selectively rescue this interaction using drugs that target ICAM, or drugs that target PCAM. And so sort of our working model of bringing it all together, like how is it that CHIP mutations are causing cardiovascular disease, is that the CHIP mutations are in the monocytes. The mutated monocytes seem to be interacting with endothelial cells differently. They're moving into the lesions, and then in the lesions, the cells themselves are pro-inflammatory. So it's not a single mechanism, but that's sort of how we think CHIP, a small fraction of cells in the blood, can cause atherosclerosis. So with that understanding, what can we actually do for patients who have clonal hematopoiesis? So you guys are cardiologists, so you guys like scores. And so one of the first things that we did in collaboration with Ben Ebert's lab was try and make a score for CHIP. So when we get these referrals from oncology, we can call them, your CHIP, we really don't have to worry about. Your CHIP, we should worry about. This is one. There have been a couple of others that have been made. These scores basically take into account information about the CHIP clone itself. So is it what mutation, what gene is mutated? Is it DNMT3A? Is it a higher risk gene like JAK2? How many mutations are there? Is it a small clone or is it a big clone? Which I told you is important. And then also some information about how the bone marrow seems to be reacting. So red cell distribution with mean corpuscular volume. If patients' bone marrows are under stress, these are indices that will change. And then things like age. And you can put it all together and then come up with some numbers and then lump people basically into low, medium, and high, because We know that although cardiologists like scores, they don't really like percentages. We've learned that from the pooled cohort equations. And so what we're able to do is identify a very small subset of patients with CHIP, probably 1% of patients with CHIP, who are actually at very high risk, and this is of dying. But same score works for developing blood cancer or developing heart disease. And then probably a larger group, maybe 10%, where we're not really sure, are you at very high risk or low risk? But 90% of patients, when they come in for a consultation, you can say, great news, you are not going to get blood cancer. You're not going to, die from this CHIP mutation. And yes, you should still take your statin, but that's for other reasons, not related to your CHIP. And so these kinds of risk stratification tools have been really helpful for patients in the clinic. This is some data that I worked on with Christy Ballantine and Pradeep Natarajan, looking at ERIC, the ERIC cohort, and showing that this same score, low, medium, high, basically equally applies to cardiovascular disease as it does blood cancer. Other interesting signals that have been, in the news recently are that patients with CHIP are actually at increased risk of developing solid cancers, and this paper picked up on that. And then also patients with CHIP seem to be at lower risk of developing Alzheimer's disease. Why that is, we're still working out, and as are many in the field. And so you actually see that patients who are at the highest risk of CHIP are at the lowest risk of dying from neurologic disease in ERIC. So save that for the question and answer or for another day. Another thing that we were able to look at is should we be, let's say a patient comes in a year later, 2 years later, 3 years later, what should we be measuring in clinic? And when we looked at the risk score, the things that were changing over time was really just the patient's age and their blood counts. That the actual clone size change, the additional mutations, mutation number, those factors seem to really only be changing if something else was also changing. And so again, when we think about resources right now, clinical grade CHIP tests are still expensive. We probably don't need to send that every 6 months, which some clinics have been doing. We probably can just follow patients' complete blood counts, and if you see a change there, then go and get a CHIP test with sequencing. Again, the other reassuring thing for patients that we found in this clinic is that if a patient has CHIP and they never develop a cytopenia, they will never go on to develop myeloid cancer. Again, never is a strong word, but effectively never. 1 in 10,000, which is basically no different than patients who don't have CHIP. And so this has been also very reassuring to patients who are sort of in this worried well category. Where we're going is building better scores. So right now, again, the score, as you notice, said you have a DNMT3A mutation or you don't. You have a high-risk gene mutation or you don't. We would like to get to the level of mutation, saying that now that we have measured CHIP in about 1.5 million people, it's not about whether you have TET2 CHIP or not. It's about whether you have TET2 CHIP, in a particular domain, or you have TET2 CHIP that is changing your methylation or changing your hydroxymethylation in some way. And so we're in our sort of clinical efforts, we're building, trying to build better risk scores and build better biomarkers for these CHIP patients. And let's say we did all of that, we would be this close to actually making a really big difference in our patients. And to get all the way there, we have to actually think about therapies. As I mentioned at the very beginning, there's a lot of interest, even stemming off of the initial papers in 2017 about looking at the inflammasome, targeting the inflammasome. We have human genetics that lines up with this. There's in vitro, in vivo animal models. There's this retrospective analysis of human clinical trial data. And so, as you might imagine, there's interest and excitement about doing this prospectively. And so that is one of 2 sort of ongoing clinical trial strategies. The obvious way to treat CHIP is if you have a mutation in IDH, you can give them an IDH inhibitor, or JAK2, you might give them a JAK inhibitor. And so those trials are ongoing, and again, all of these have started in the last probably 2 or 3 years. So I can't tell you which ones work, but I think we'll hear data very soon on several of these. And then the other approach is using canakinumab or using sort of the next generation of canakinumab. So this last study is sponsored by Novartis. The DFV-890 is an oral medicine that's an NLRP3 inhibitor. MAS825 is basically canakinumab. So it's like canakinumab, the next generation. It's an IL-1 beta antibody with an IL-18 antibody. And again, why? Who knows? We'll find out. This last trial we recruited for at Vanderbilt, it's all done, so we should have data, at some point in the next probably 6 to 12 months about what happened. And again, this last one was just a biomarker study. I think one of the other sort of interesting opportunities in CHIP is how do we think about all the different diseases that patients with CHIP are getting. So the investigators who are doing the IDH trials are reporting that patients who have IDH in their trials, are getting better in terms of their autoimmune disease, and that's not part of the trial. That's not part of the outcome. They're focused on the cytopenia. So, how do you come up with some kind of composite endpoint that is improvement across, a number of different health factors? Yeah, and just some more details. Again, Brett Heinlich in my lab with Aaron Addy, were our site investigators for this one. And so it was just really a simple 12-week study focused on biomarkers of inflammation that are, in some sense, validated now that the CANTOS study has sort of linked reduction of inflammation to reduction in outcome. And so I'll end with this. Eric Topol a few years ago said that the real problem we have with CHIP is actually a marketing problem, which is what you would expect from an influencer like Eric Topol. He said that really what we need to do is drop the word indeterminate. And if we just called it clonal hematopoiesis, then people would take it a lot more seriously than if we tell the oncologist that's of indeterminate potential. So, the rebranding is ongoing. And lastly, I want to thank my lab, thank Brett. Again, Michael did a lot of this work with Tara. We had a great dinner here in LA when ASHG, the Human Genetics meeting, was here a few years ago, and wonderful collaborators, and just a few of whom are up here. So thank you, guys. Yeah, of course.
So does having a chip mutation anything look prone to developing another chip mutation because of the clonal expansion, or not?
Yeah, it's a great question. The answer is yes, right? So some number of these mutations are just the more cell divisions you have, the more likely you are to get a random mutation. But what also happens is there's synergy. So if you have a TET2 mutation, your likelihood of then developing an IDH mutation or developing like factor mutation goes up because those two together have more of a proliferative advantage than just one. So yes, if you have one, you're at increased risk of getting another.
Patients that have like clonal regression that were maybe not on like anti-inflammatory drugs or something, are they protected against certain diseases?
It's a really good question. We haven't had sort of this full size to really understand. Like, we've looked at it as like a continuous variable that if your degree of growth is faster or degree of reduction is slower, then you are protected from sort of a laundry list of diseases. But it's a great idea to go back and look at specifically the ones whose clones were maybe stagnant or shrinking. About what happened.
Great talk, Alex. Amongst my many questions, your PACER slides were very interesting, and I was wondering in those analysis of you did if kind of like the sort of again the ring sort of analogy, like were these samples you all looked at one point in time, or were you looking at these samples longitudinally to look at this timeline of clonal expansion? My second question to that is, did you see thresholds where people, I don't know if you looked at phenotypic, like these patients had already manifested cancer or heart disease. Do you see a certain threshold along this timeline? You saw an increased risk.
Yeah, great question. So all the PACER analysis, what's really cool is that you're doing it one time. You're basically doing it, you think about it as you've just cut down the tree and now you're counting how many rings it had. You could do it serially, but if you had serial measurements, you would just measure how big the clone was and actually know what the growth rate is instead of having to estimate it. I think, in terms of, are there thresholds for growth rate that seem to really be particularly bad? I think that's similar to the last question. The numbers of studies that have multiple blood samples and have detailed sort of outcomes is far too few, and has been really a challenge. And so what we've been trying to do there, going back to the cell-free DNA question, is that all of these studies, even if they have one blood sample, they have lots of plasma samples, because everybody knows that the plasma changes over time. And so we developed, the hard part is getting the DNA out of the cell-free DNA, but I think we found a way to do that. And when we do that and put it on a normal chip assay, we think that we can measure sort of growth serially. So I think putting those two things together may be the solution to this question.
I found the fat biopsy thing really interesting. Have you looked at other like tissue samples, either from, biopsies from cancer, CSF, lymphoid tissue?
Yeah, so the solid tumor cancer is super cool. And again, it's the cardiology audience. So I think you'll find it cool, even though you're cardiology. So patients who have CHIP have worse cancer outcomes. And the question is often been why. There was, we did an analysis which was published in like Cell Reports Medicine, and then a month later, another group did the same analysis and was published in the New England Journal of Medicine. So that was probably poor planning on our part, I guess. But what we found is that if you have a CHIP mutation in the cancer, those were the people who did worse. If it was just in the blood, it didn't really matter. And so there is something about, and I think it's the same story with the plaques, where just the fact that these mutated macrophages have invaded the tissue, they're causing damage in the tissue.
So what about atherectomy specimens, heart specimens? You must have access to some.
Yeah, I think we're running in many different directions with this. The atherectomy samples are hard to process, and again, look forward to people's guidance this afternoon on how to work with them. Sid Jaiswal has been doing that in collaboration with this Munich biobank that has, I guess, a bunch of these atherectomy samples. And again, just getting cells out of them, I think, has been challenging. We've been really interested in kidney disease, in part because around the same time Michael was there was a visiting student from Canada who was really interested in kidney disease, and so now we have an R01 on kidney disease. It's like, and so you're able to find the same mutations that have invaded the kidney, and that seems to increase risk of, if you have kidney injury, of having delayed recovery from kidney injury, and some of the same inflammatory pathways. So now we're trying to do more of this. But yeah, I think there's a lot of opportunity to look in tissues. And then your colleague at UCSF with autopsy studies, has been picking up the.
Yes.
We've been helping them with the sequencing. I'm not sure what's happened.
A couple of questions. One is there any
People should feel free to get back to their clinical work.
other factor beyond genetics and environmental rate, diet, population that
It's a risk factor for kids. It's a relatively weak risk factor. It seems to increase ASXL9, one of the CHIP genes, more so than some of the others. We've looked at radon exposure, like from the and so we all have these like radon mitigation systems. They're again weak signals, but probably our quality of measuring the exposure is probably the best. And anything that can cause a mutation can cause CHIP. But we're also very interested in trying to learn more about what causes that CHIP clone to grow. That's where air pollution is. I don't have answers there.
And the second question, I don't know if I misunderstood one of your slides, but you put some data that the inhibition of interleukin-1 beta increased cardiovascular disease. My understanding is that
The opposite.
yeah, okay, so I definitely misunderstood.
Like a big question, but the Alzheimer's, can you just comment on?
Yeah, there's two thoughts in the field. One is that it's just an epidemiology artifact because patients who have CHIP die of something else and don't live long enough to get Alzheimer's. So there's one group of people that believe that. There's a different group of people that believe that the TET2 mutations are in the blood. Microglia are derived from blood or blood stem cells. They're the macrophages of the brain, and that those microglia are sort of like super microglia, and they do a better job of eating plaques. And so there are several mouse studies. Sid Jaiswal led one of them. Catherine King at Baylor has led another that have shown that's true in mice. But there's a lot of interest because on balance you still don't want CHIP. It increases your risk of dying, increases your risk of heart disease, whatever. But maybe that tells us something about how to develop like a cellular therapy for Alzheimer's, which would be wild.
How much does a chip cost?
In our lab.
Yeah, you brought up the plasma because Atlas, for example, here, people are coming back and getting blood samples and plasma sitting there. How much does it cost to do it?
Yeah, it's like between $10 and $20 a sample.
You have to isolate the.
You have to isolate the DNA,
That's
which is
expensive.
Yeah.
How much plasma do you need? Like 1 ml?
Yeah, one ml is fine, and the cost to do cell-free DNA isolations is probably $1 sample range is my guess. To do it off of blood is a lot cheaper.
You could enrich too with the score, using the score without the DNMT3A. You could just go through the clinical data and pick out 100 or 200 people with it.
When you start with the blood, you screen with your blood samples from the data that you're getting back from Regeneron, and then you plasma in the samples.
Paul. Julia, I have one more question.
So one of your first slides, you had that the incidence of CHIP by gene, and I think it was DNMT3A and TET2 are the highest incidence. But aren't those also the most dangerous? And isn't that kind of the opposite of what we how we evaluate variants in the genome? Rare variants are typically the more dangerous or disease causing. I mean, I'm making a generalization. Why is it that bad CHIP mutations have a higher incidence?
I think I know what you're talking about. You're talking about natural selection.
Yeah.
It's hard to think from a lens of a germline genetics person about cancer genetics because it's exactly the opposite.
Okay.
So in the cancer world, if something is pathogenic or has under strong selection, it rises to the top because it's out competing. Whereas in germline genetics, if something is bad, then it gets selected again, so it goes away.
And that graph kind of takes into consideration the size of the clones too.
No. It was just how often you find it.
How often you find it. Okay.
And there's split CHIP, but frequency of each one.
Yeah. Well, thank you so much, Alex.